First and foremost, I would like to apologize for not being there in class today, but I appreciate everyone for being flexible and able to watch this presentation in multiple formats. So thanks, Josh, for the beginning half of the presentation. Now we're going to talk about the amphipod feeding habits. As discussed earlier, these guys are scavenger organisms. They will eat basically any type of organic material, whether it be zooplankton, phytoplankton, or detritus, decomposing material of any sort, really. And they specifically use their legs to create a current in order for the food to reach their gnathopods, which they then use to bring the food up into their mouths. As for their reproduction habits, unlike a lot of marine organisms we have covered in this class, these guys only reproduce sexually. They do not show hermaphrodism at all, and they do not produce asexually, only sexual reproduction. And the specific production of offspring is ovoviviparous, meaning they do have eggs, but they stay within the mother and end up hatching live young. So their breeding season is typically between March and October, and this is mostly dictated by the molting schedule of the females, because the molts are dependent on water temperature. So that said, the water needs to be warm enough, which is why the breeding season is typically in those months. Another note is that gammarids are monogamous and only experience one mating season per lifespan. That said, though, they can copulate multiple times per breeding season with their chosen monogamous partner. These guys do have a specific mating ritual, which, as per usual, starts with the males performing a mating dance to attract a female. Once a female is attracted, the males will actually mount the dorsal side of the female and use their legs to kind of feel around their back and their exoskeleton. And this kind of begs the question of if females have external pheromones. Studies have shown that there have been males that have felt the back of the female's exoskeleton and have then ditched their partner after attracting them, which kind of, like I said, we can hypothesize from that the females have external pheromones kind of signaling if and when they are ready to actually mate. So once they feel the female and get the signal that everything is good and ready to go, they use their gnathopods to hold on to the female, and they kind of just grip onto her. They will let go briefly for her to molt, and then will quickly reattach and mate directly thereafter. So she has to molt before the mating can actually begin. Once she molts, the male uses their back legs, otherwise known as uropods, to feel the female's underside for the oostogytes, which is seen in this bottom picture, labeled G and H over here. And those are just the flaps and a structure that kind of contains all the eggs underneath her belly side. Once the male feels those, then he releases the sperm, and the females use her pleopods, which is the more middle of the body legs, to navigate that sperm to her brood pouch where the eggs will be fertilized. Like I said earlier, the gammarid life cycle is temperature dependent. This is because the molts are temperature dependent, and the molts are what separate all of the life stages, specifically called instars. An amphipod can undergo anywhere between 15 to 20 molts in a lifetime. Typically, the average molt when mating begins is when the female molts into her ninth instar. The fertilized eggs will remain in the pouch and hatch after 1 to 3 weeks. The hatched young will stay in the brood pouch for another week, typically, and that's until the mother molts again. So the eggs and young are usually staying in the female's mother's brood pouch for 2 to 4 weeks. After that mother molts again, like I said, the young become independent and exit the pouch, and they are still considered juveniles during their first 6 molts. And after their 7th molt and into their 7th instar, they are considered young adults, because that is when they start to show sexual dimorphism, and you can kind of determine gender after that 7th molt. This entire life cycle is typically completed within a year on average. As for the specifications of how to culture these amphipods, it should be easy. We seem to have had some difficulty with ours, but what you would want to do is obtain a starter culture from the manufacturer, place that culture in a minimum 10-gallon tank. That's what we did in ours, and it has to have adequate air circulation, and then you just feed them every other day, if not daily. They don't have to be fed daily. Every other day is fine, but as long as they're fed, that should be good. And they do have a diverse diet, so they can be fed a whole bunch of things, which includes spirulina powder, which is kind of like a blended pulverized algae. They can also eat pulverized fish flakes and fish pellets, as well as just natural growing algae in the tank, etc. Like I said, they're very omnivorous and can eat a wide variety of things. One thing to note though, the water should be around room temperature, anywhere between 72 and 82 degrees Fahrenheit. This is important because they are cold-blooded, so naturally the warmer temperatures will allow for more active feeding and reproduction. And like I said earlier, the water temperature is kind of the main name of the game when it comes to when and where they will molt. So in order for them to complete their entire life cycle, the water has to be warm enough for the molts to occur. And the main takeaway from this slide is just that as long as you've got these minor things in consideration, culturing amphipods is quite easy since they are generalists. Lastly, I just kind of wanted to bring up the idea of taxis, and specifically Gamma-Rid taxis, since this will be a portion of our lab activity later on. But just to define the word, taxis is the directional movement in response to a certain stimuli. And you can see in this chart here, there are all sorts of types of taxis. There are going to be taxis regarding food, which is called trophotaxis, taxis with light, which is phototaxis. You can have taxis with gravity or magnetism. You can have magnetotaxis. There's all sorts of different things you can measure regarding the directional movement. The main thing, though, is that these taxis can be described as positive or negative. So a positive taxis is that the organism moves towards the stimulus, and a negative being the vice versa, an organism moving away from the stimulus. Like most organisms, Gamma-Rids exhibit a positive trophotaxis, meaning they move towards food, which is good. And the phototaxis is what we will be checking out in the lab today, which is regarding their response and movement regarding light. Here are our references that we used to make this presentation. And lastly, does anyone have any questions for Josh? Sorry I couldn't be there, but you can pause the video now if you guys have any questions. Okay, so for our lab activity today, we'll be giving you guys some samples from our tank to observe under the dissection microscope. So first and foremost, the initial thing you have to do with these samples is to be able to distinguish the Gamma-Rid species, which are our amphipods, from the other organisms in the sample. Because it's likely that we'll have a lot more than just the amphipods in our sample in our tank. We've got lots of algae. I think we even have some worms in there. So Use your knowledge based off of this presentation today to kind of morphologically determine which ones are the amphipods and which ones aren't. Then once you feel comfortable and confident after doing that, then you would observe and take notes on the phototaxis behavior of our amphipods. So originally we were going to use note cards, but I believe Professor was able to kind of make our own phototaxis simulating petri dishes. So you can use those. There will be a distinctly dark half and a distinctly light half. And just kind of observe them, take notes, and see if they exhibit positive or negative phototaxis. And that's all I got for you guys today. Thank you.